Hi everyone, it's Mr. Sinti, and today I'd like to discuss the most magnificent adaptations that prokaryotes have that make them very successful. And one of the things that I want to sort of emphasize right out of the gate about these prokaryotes as being masters of adaptation is you got to expect that. I mean, these are the first organisms on the earth, and they've been with us since the creation of life, and they're totally dominating. They're the most numerous, and we're going to see in this particular video. I hope you enjoy it. That they have these incredible adaptations, which are traits that allow them to be so successful. And so, one of the things that gets my attention is when I look at this picture right here of Salt Lake City, not Salt Lake City in particular, but the Great Salt Lake in Utah. Do you notice how it's kind of like pinkish in coloration right here? This pink color comes from all the prokaryotes or bacteria that can live in here. And you're like, how can bacteria live in here? Check this out. The salt concentration is 32%. And if you're not impressed by that, let me sort of refresh you on salt and saline concentration. The ocean is 3.5%. 3.5%. This is 32%. This is really, really salty. Now 3.5% is like what, 35 grams in a liter of salt. So this is like, a lot of salt. It's like, how can anything survive in this? Because salt being an ion, not to get into detail with this, but the sodium ion, chlorine ion can be very disruptive to protein shape, etc. It's really a killer. But there are some bacteria called halophiles that actually can survive in these extreme saline conditions. And so that's a pretty good adaptation, don't you think? And so prokaryotes thrive almost everywhere on the earth, including places where other organisms can't handle it, places that are too acidic for most organisms, too salty, as we were just talking about, too cold, or even too hot. And an example of that is beautiful hot springs in Yellowstone National Park in the United States. This is really hot. Normally, organisms cannot handle that. The heat denatures proteins in particular. It's not suitable. But there are prokaryotes that can survive in these harsh conditions. And one of the organisms in particular that is a real popular one for bacteriologists and also microbiologists is Thermus aquaticus, because we've been able to isolate their DNA polymerase, which is what we call TAC, which is sort of an acronym for Thermus aquaticus, TAC polymerase, and it's sort of heat stable. And so we use that, if you're familiar with this, we use this particular polymerase in PCR in order to amplify genes. It was one of the great discoveries in biotechnology, so that's pretty cool. So bacteria can even handle these hot environments. They can even handle acidic environments. So this Helicobacter pylori bacteria right here can actually thrive in the stomach. And I tell you, there's not a lot of bacteria that can do that. Usually the stomach is so acidic that it helps us so we don't get sick from eating food that are just totally laced with bacteria. We believe that this particular bacteria is one of the major causal agents of ulcers right here. And so you can treat ulcers in some instances with antibiotics that would then kill these guys and help you out there. So one of the things I want to say is prokaryotes are, if you didn't know this, are microscopic. They're very small. They're often single-celled, but they can live in colonies. And what they lack in size, however, they make up in terms of numbers. And so they're kind of fearsome that way in terms of numbers. And I'll explain how that's an advantage or an adaptation. There's so many of them that it's hard to even imagine. There's more bacteria in a sort of a handful of soil than people have ever lived on the earth. Okay, so numerous. And they take on interesting shapes. Like these are particularly round. The ones that we saw right here were sort of rod shaped like that. And some of them are kind of spiral in shape. And so one of the things about their appearance or their phenotype was how biologists classified them for centuries. And up until most recently. And so systematics is the study of the relationships between organisms and other organisms, evolutionary trees, phylogenetic trees, collatographs, these kinds of things. And so lately, our molecular understanding of DNA sequence has been able to better classify these prokaryotes appropriately, as opposed to just how they look, which is not always a great way of determining who's more closely related. And so when we apply molecular systematics, in other words, our molecular data to better understand relationships between organisms, we're able to now understand that in terms of all life right here, so all life can be broken up into what we call 3 domains. And one of them is the eukaryotic domain, and that's a cell, if you're not familiar with it, possesses a nucleus. But the vast majority of organisms on the earth are broken up into the domain archaea, which are prokaryotes, okay? So not possessing a nucleus, that are extremophils that really handle those harsh conditions that I was describing. And then there's the bacterial world. And what's interesting about this is that, both archaea and bacteria are prokaryotes, but I love this, that new DNA sequencing has suggested that archaea is more closely related to eukaryotes like ourselves than to bacteria. How about that? That's kind of wild. And so the use of polymerase chain reaction, in other words, like amplifying DNA sequences and then ultimately sequencing them, has led to our understanding of molecular relationships between them. And so again, I want to talk about in this video some of the structural adaptations that have allowed bacteria or prokaryotes in general to be successful, some of their metabolic adaptations, and some of the relationships they develop with other prokaryotes and with other organisms that make them successful. But right at the start, let's talk about structural. So again, the shapes, they could be circular or spherical or rod shape or spiral. One of their more important structural adaptations is the fact that they have this cell wall. And an organism that has a cell wall is not only protected from outside environment, but really what it's doing, it's protecting the organism from bursting in a hypotonic environment. And that's really useful because water enters into cells and it has a tendency to cause them to burst. But because they have a cell wall, they don't often burst in a hypotonic environment. But one thing I can say about the cell wall of prokaryotes is that it's a little different than what you might be thinking in terms of a eukaryotic cell wall. Plant cells and fungi have cell walls, but theirs are made up of the sugar cellulose or chitin, which is this sort of polysaccharide nitrogen compound structure. But the bacterial cell walls are made up of peptidoglycan. Now, peptidoglycan is a combination also of sugar polymers and some proteins, and so it's just simply shown here, and it's on the outside, in this case, of the cell membrane. And so their bacteria can be classified either by how they stain, and that, again, that's been with us for a long time. So if you treat bacteria to a variety of stains, in particular this crystal violet, if the bacteria pick up the crystal violet and the peptidoglycan does that, the bacteria appear kind of purplish. And so those are called gram-positive bacteria, and you can see the gram-positive ones in purple. Now the gram-negative ones have a thinner peptidoglycan layer, and that's sort of sandwiched in between 2 layers of membrane, and so they do not stain purple, and they kind of pick up this red dye that usually washes away. So they're kind of the pinkish ones are gram-negative and the purple ones are gram-positive. So the point of this is antibiotics, one of the ways in which, there's a few ways, but one of the way in which antibiotics, things that kill bacteria, they actually target the peptidoglycan wall. And so they're often more effective in the gram-positive ones. So what they can do is punch holes in the peptidoglycan cell wall, and then simply water comes in and then kills the germ. And so gram-negative ones are likely to be more resistant to antibiotics. And so you might be familiar with this. Some bacteria also can possess a sugary coat, a polysaccharide or protein layer called a capsule that covers some of the bacteria. You might be familiar with this in the classic historically significant experiments of Dr. Griffith when he was looking at pneumococcus bacteria in mice. He noticed that one strain had this smooth capsule coat on the outside, which was actually caused pneumonia in mice, and ones that did not possess the capsule were destroyed by the immune system of the mouse. And so this capsule can be a useful adaptation to survive if you're a bacteria. As it turns out, white blood cells, especially the ones that are macrophages, like a neutrophil, comes along and has a very difficult time grabbing this because it's kind of like slippery, if you will. But it also, because of its sugary nature, it can also be adhesive to tissue, which makes it particularly dangerous, but again, advantageous for bacteria. So here's a capsule that you can see right here, the sugary sticky coat that's adhering to a tonsil cell in the back of the throat right there. And so that's not so good. And so some bacteria have these thread-like structures. Let me just show you a picture of that when I'm trying to describe. So here's the bacterial cell. They have these filaments that extend from them. And so these filaments come in two kinds. They're either called fimbriae, which allow them to stick to substances or other bacteria in a colony, or these pili, which are sort of hollow-like extensions from the cell that allow them to exchange genetic material, which is also a tremendous adaptation. If you can share or recombine your DNA between organisms, your offspring will be genetically different, and therefore that's an advantage in an ever-changing environment. But this fimbriae is important too, because these numerous fimbriae can enable a bacteria to attach to surfaces, which are useful to them. Now, mobility, one can say, well, gee, mobility is an important adaptation for all living things. And yes, I would agree with that. But did you know that bacteria have mobility? And how they do that is generally with a flagella, which is a protein extension that allows them to move around, sort of like the human gamete male cell, the sperm. And so in a heterogeneous environment, in other words, an environment that is like really good in one area and maybe not so good in another area, the bacteria are capable of movement, you ready for this, toward the good area. And so we call that type of orientated movement taxis. In other words, a directed motion toward a positive stimulus or away from a negative stimulus. So in other words, if you put glucose in this area, they would move toward it because they want that as a source of energy. And so this is what you'd call a chemical, chemotaxis, moving toward a good chemical like glucose or away from something dangerous. And so most bacteria propel themselves by these flagella. Some have one flagella, some have on both sides, some have many, as you can see here. What's kind of interesting is that I mentioned that the human sperm has a flagella. There are also protists that's also in the domain Eukarya, in the phylum Protista. So this is distant from archaea and bacteria and protists. The proteins that, when we look at the proteins of flagella, they're often remarkably different, which suggests that these flagella evolved independently from one another, that they're not necessarily a consequence of ancestral descent with modification, but it's something that has arisen on their own and evolved independently. That's kind of interesting. Now, their internal structure and their DNA is also an adaptation. You're like, well, what do you mean by that? Well, usually prokaryotes are fairly simple. They possess ribosomes, but they don't really possess any internal compartmentally membrane possessing organelles. But what they do is that they have specialized membranes that perform metabolic functions. And the way that they enhance this is that they use their outer plasma membrane and use infolds. In other words, convolutions of the plasma membrane can assist them. And so that's sort of an adaptation, if you will. So more surface area in this bacteria allows them to better take up oxygen from the outside. And in this photosynthetic prokaryote, it allows them to produce, have more chlorophyll pigment embedded in these membranes, thylakoid membranes, which enables them to maximize photosynthetic rate. So that's kind of a structural membrane adaptation. Also, speaking of DNA, obviously a prokaryote has less DNA than a eukaryote, many less genes. And if you didn't know, the genome of a bacteria is in one chromosome, usually a large circular singular chromosome. But they also have these small, so a big chromosome like this, but they also have these circular rings of DNA called plasmids, and these are rather useful. What genes are contained on these plasmids? Some of them, called F plasmids, contain genes for the pili, which allow them to genetically exchange DNA through conjugation. Some of them are R plasmids, which contain genes for resistance to antibiotics. So that's clearly an adaptation. Okay, so here's a better picture of that. Here's the chromosome of a bacteria sort of busted out right here, and here's some circular rings called plasmids. And again, those are very useful. And again, to emphasize it again, is that these R plasmids carry genes for antibiotic resistance. And so antibiotics kill bacteria that don't have resistance, obviously. And so if you can exchange your plasmids, then more bacteria will be resistant. And then through natural selection, one of our concerns is, we thought we had conquered bacteria when we sort of discovered antibiotics, but the more you treat bacteria with antibiotics, the more you're basically going to select for the bacteria that are resistant, and eventually they'll become more common. And that's the way natural selection works. And so there's becoming a concern from the human perspective. This is good for bacteria. From a human perspective, this is not so good, that they're becoming more resistant to antibiotics. Now, one of the most important adaptations that bacteria have is their ability to reproduce. And so this is the heart of this particular video. So if there's any come away, take home message on this is what I want you to get the most, which is, if you didn't know this, bacteria divide very quickly. So under ideal conditions, warm, lots of food, no competition, they can divide very rapidly, like even 30 minutes in some instances. But we'll just say every couple of hours they're dividing. Now, they divide through binary fission. So in other words, one cell will simply divide itself into two cells. Now, this is cloning or asexual reproduction. And so it's very efficient in a sense that for every one cell you get two cells. And then you get four. And so there's this exponential growth. And so that, that's very useful, asexual. But one of the drawbacks to asexual, if there is one, is the fact that these are clones. They're genetically the same. And so that puts the colony or the population in danger if there's some kind of environmental change. However, because they have a short generation time, this allows them to reproduce rather rapidly. Okay, and you're like, well, all right. So in other words, they're dividing binary fission, and they're doing it rather quickly. So what I'm saying is that, to summarize this, is that the number of bacteria, the number of them, can go up rather quickly. And you're like, well, okay, so that's kind of an adaptation, but what do you mean by that? Well, as it turns out, the fact that they're able to divide so quickly allows them to evolve more quickly and change more quickly. Because, and I'm going to explain this kind of slowly. And so what we've seen in terms of laboratory experimentation is that, if you take a bacterial colony and you're growing it, like for example in a test tube like this in a nutrient growth medium, and then you take a small sample of that and then transfer it over to a fresh nutrient medium, you can keep growing this bacteria for a long period of time. And what we've seen is that the bacteria change, they adapt, and they're better than they were before. And like, what do you mean by that? Well, though bacteria are not primitive but highly evolved. Sometimes we get the impression that we are highly evolved and the bacteria are primitive, but in fact the bacteria can evolve as well. And so what I'm getting at is, when you grow these bacteria, and you can keep the original colony, but when you keep transferring them over and over, generations and generations, these new ones can have changes occur. And you're like, well, how can changes occur? You might know this already, spoiler alert. Through mutation. A change in the genetic material will allow them to alter their phenotypes. And so therefore, when you put these future colonies against, like in a competitive environment, with the original strain, you can see that they grow better. So they're better adapted than they were before. And so it shows that they're capable of improving, if you will. And so how is this occurring? Well, it's because they're rapid reproduction, but mutation is what's doing this, and genetic recombination. So let me slow it down for a second. So prokaryotes do have considerable genetic variation, but it's like, well, how does that occur? Well, their rapid reproduction is one thing, so they're able to spread that. But most importantly is its mutation is what changes the DNA, okay? But even though the mutation rate is not very high, the fact that they reproduce very rapidly is what causes the genetic diversity. And then the third reason is genetic recombination. And I can mention this. I've created a whole video on just this concept of genetic recombination in bacteria, which is transformation, which is the transfer of genetic material between one organism and another, conjugation, which is exchange of genetic material through the pili, and transduction, which is viruses, phages, transferring genetic material between bacteria. But I want to stay with this mutation rapid reproduction for a moment, if you don't mind. This rapid reproduction and mutation is a critical idea. I really think that this is such an important heart of why bacteria are so successful on the earth. It's the bacteria reproduce through binary fission, and their offspring are genetically or generally identical because this is asexual. But through mutation, that does cause changes in the next generation because the DNA sequences are different. But the mutation rates are generally kind of low, but because of the rapid reproduction, the mutations accumulate in the population immensely. So thus, the high diversity that you see in the prokaryotic world, the genetic diversity from the mutation, allows for this rapid evolution. Because of the diversity, when things change in terms of an environment, it'll select those phenotypes that are better suited, and those are therefore more successful. So their genetic variation is the key to their success. And again, just to repeat, their genetic variation is the key to their success, and that comes from mutation. But even though the mutation rate is kind of slower, they're still able to reproduce so rapidly that it results in a diverse population. And then again, the diverse population is even further enhanced because bacteria can recombine their DNA, which causes diversity. And they can do that either through transformation, transduction, and conjugation, these three ways. Okay, and so that's called horizontal gene transfer between bacteria. So that's important. This is how we do it in terms of like sexual reproduction, is we rely on the recombination between two different parents to make our offspring different, and that allows us to be adaptive because our generation times are much slower than bacteria. Now, another kind of a cool thing about reproduction, before we close the book on that, I find this to be particularly curious. Like a bacteria, it's like it can remain dormant for a long, long time, like centuries long. And when the environment like goes sour for the bacteria, it doesn't totally kill them. It can make them inactive. And as it results in, there's this structure that some bacteria have called an endospore. Let me show you a picture of this. So inside the bacteria, there's this endospore. It's not truly a spore by the definition of this. It's it's kind of like just the heart and soul of the bacteria. So the genetic material is in here. So under harsh conditions, the whole like cytosol of the bacteria can be worn away, and just the endospore remains. And as the endospore remains, it's sort of a stripped down, dormant version of the bacteria, which can reproduce in the future when the environment is a little bit better. And so bacteria can really hang out for a long time and not totally die. We've found endospores in in mummies, Egyptian mummies that have been in that where you'd think that nothing can survive that for centuries. It's kind of interesting. So let's talk a little bit about metabolic adaptations that bacteria have that enable them to be successful. And in two particular things I want to talk about is oxygen and nitrogen, and how they're able to handle that metabolically. Did you know that the bacteria are so diverse that there's actually some bacteria that are sort of obligate anaerobes, meaning that they require oxygen for cell respiration, and without oxygen, like us, they would die. But then there's some obligate anaerobes. In other words, they're poisoned by oxygen. So they're anaerobes. So they respire just through fermentation to get their energy. And so oxygen would kill them. Isn't that incredible? So they're able to handle oxygen environments and actually anaerobic environments, like deeper in the soil, for example, or higher in the atmosphere. And then there's some bacteria that are facultative. In other words, they can go both ways. They can survive with or without oxygen. So that this allows them to be successful in living in a variety of different places. And then nitrogen, look at this. Like they're able to, as you know, nitrogen, I can go off on nitrogen. I feel that nitrogen's very significant as it's a component of amino acids and nucleotides, DNA, RNA, and proteins. So prokaryotes can metabolize nitrogen in many different ways, in more than we can, if you will. And in one perfectly good example of this is nitrogen fixation. Some bacteria, nitrogen fixing bacteria, can actually grab atmospheric nitrogen and convert it into ammonia, which is really useful in terms of acquiring nitrogen to make amino acids and therefore survive. So that's critical. And then you got to love this, like bacteria can even be cooperative, not only within themselves or between cells. So take a look at this. In cyanobacteria, so blue-green algae as it's sometimes referred, but it's really a bacteria, they're capable of photosynthesizing, in other words, making their own sugar, but they have a specialized cell called a heterocyst that looks like this. This heterocyst that's capable of nitrogen fixing. So this is able to get a good source of nitrogen from the air and produce ammonia, which is then converted into amino acids. And that is useful to the photosynthetic cells, which are mainly responsible for producing sugar. So together, they're very strong. And so they exchange their good qualities amongst each other. So it's sort of a metabolic cooperation. I find that to be particularly interesting. And then finally, there's cooperation between bacterial cells. And so one great example of this is something called a biofilm. So a biofilm, it isn't restricted just to prokaryotes, but basically it's any microorganisms that sort of stick to each other and form a colony, if you will, especially on the surface. It could be on the surface of another living thing, or it could be something like plastic or the tabletop, and for example in a hospital, which is a little disconcerting. But these biofilms, like groups of bacteria that are attached to the surface of a non-living surface, can be useful. And so what an example of what I mean by that is a bacteria living in sort of this colony or biofilm usually has significantly different properties than a free-floating bacteria of the same species, because the dense population usually protects them from the environment. In other words, the ones that are sort of on the inside of the biofilm are protected from some harsh environments. Like, for example, detergents, which usually destroy microorganisms by messing up their membranes. The ones inside are protected from that, or they could be protected from antibiotics when they're living in a biofilm. And so living in this sort of matrix protects the inner part of the community. I find that to be kind of interesting. And then another way bacteria are sort of adaptive is that they're able to communicate between each other. Now, like, whoa, what do you mean? Well, they have a system of producing chemicals called autoinducers. And so when the number of bacteria are very small and they produce these chemicals, sort of like a hormone, the other ones don't pick them up because the autoinducers sort of diffuse. But when there's enough bacteria producing these autoinducers, they can pick up the chemical message, and therefore they can sense that the number of bacteria is very high. And so it's a way that many bacterial species, and it's called quorum sensing, because when they develop a quorum, in other words, a large enough number, they can start to coordinate activities, because these chemical messengers can actually cause gene expression to take place. In other words, the bacteria can all start doing something. They could start secreting a toxic chemical, or they could be producing bioluminescence, but only when their numbers are high. And so it's a coordinated event. And so it's kind of a decision-making process when you have a decentralized system, because individually they can't really accomplish much. But it's a means of bacteria assessing the number of organisms when that number gets really large. And then when it reaches a certain threshold, they could start to do something that they weren't doing before. In other words, producing a biofilm, which is kind of a complicated thing. I'm sort of dodging around this, but if you were interested in how biofilms form, I welcome you to research this a little bit more. But basically, it allows them to be successful if they form a biofilm. And again, some of the constituent chemicals that are involved in the adherence of cells to one another in a colony in a biofilm are only produced when the number of bacteria are at a higher number, and they do that through quorum sensing. Okay, so cell-to-cell communication, or quorum sensing has sort of evolved in the sense that the consequence is producing the biofilm. And so that's an adaptation to allow bacteria to survive. So through communication, through rapid reproduction and mutation, through structural adaptations and metabolic adaptations, prokaryotes dominate the earth and are some of the most essential life forms. It's hard to imagine that life could even exist on the earth without them. So I hope you learned something, and I hope you enjoyed, or even more appreciate, the numerous adaptations of prokaryotes, and they truly are the masters of adaptation. Thanks for watching.